Today we're going to look at Mendel and the gene idea. Originally, there was a blending hypothesis around in the 1800s where they thought the genetic material was the mix of two parents and that the traits would blend together similar to the way paint colors do. The particulate hypothesis is closer to the gene idea, where the parents would pass discrete inheritable units that would retain their separate identities in the offspring. So the traits would be passed from the parents, and the offspring would inherit one or the other trait that would be manifested in the offspring. Mendel used the scientific approach to identify two laws of inheritance. The character is a heritable feature that varies among individuals, such as flower color, and the trait is the variant for a color. So the trait would be either purple or white flowers in the example we're going to use below. Mendel chose to track only characters that occurred in two distinct alternative forms, and he chose pea plants because of their ease of growing them and their quick generation times. A plant that is true breeding is a variety that would produce only the same variety as the parent plant after self-pollination in successive generations. So if you continued to self-pollinate the purple flowers, you would just continue to get plants with purple flowers. If you continued to self-pollinate the ones with white flowers, you would continue to get ones with white flowers. With hybridization, you have a mating or a crossing of two contrasting true breeding plants. That would be mating the purple flowers to the white flowers. Your P generation is the parental generation. This would be the two true breeding plants. In the F1 generation, which is your first filial generation, filial just means son, the hybrid offspring would be the result of crossing two true breeding parents. And in this case, all of them had purple flowers. In the F2 generation, you would self or cross-pollinate hybrids within the same generation, and what we saw was about a 3-to-1 ratio. You would see the white flowers come back again. That told us that the blending model was not correct. If it was, you would have seen a cross between purple and white flowered plants produce pale purple instead. So here you saw the white flower trait reappear. The dominant versus recessive trait occurred in about a 3-to-1 ratio in the F2 generation. And Mendel used this information to deduce the law of segregation. The dominant trait is going to be the one that is expressed or seen if it is present. The recessive trait will only be seen in the individual if they have two copies of the same recessive allele. The alleles are the alternative versions of a gene. Alternative versions of genes are going to account for the variations we see in the inherited characteristics. So for each character, an organism will inherit two copies of the gene, one from the mother and one from the father. If the two alleles in a locus differ, then the dominant allele is going to determine the appearance, and the recessive allele will have no effect on the organism's appearance. The two alleles for a heritable character are going to segregate during gamete formation and end up in separate gametes. This would make sense since the genes are actually on the chromosomes, and the chromosomes are what gets spread into the gametes. Here we look at the same example again, and we're able to see the genetic makeup of the gametes. In our P generation, we've got the purple flowers that are going to have two capital P's. Capital letters are used for the dominant trait, small letters are used for the recessive trait. For the white flowers, they would have two small p's. They're true breeding, so they're going to have both alleles be exactly the same. With the purple flowers, the only type of gamete they can produce are going to have the big P. For the white flowers, the only type of gamete they can produce is the small p. When we get to the F1 generation, all of those are going to be purple flowers that are going to have one allele from each parent, so a big P and a small p. When those flowers go to make their gametes, half are going to get a big P, half are going to get a small p. When we put this together and look at the 3-to-1 ratio that you get in the F2 generation, we can use this Punnett square here, which is a diagrammatic device that allows you to predict the allele composition of the offspring, as long as you know the genetic makeup. Here we have a big P and a small p for the gametes, the eggs along the side, the sperm are lined up across the top, a big P and a small p. we bring all of the letters down into the boxes below. Big P's are brought down into the box together. A big P and a small p brought down to the box together. A big P and a small p brought into this box, and two small p's into the fourth box. The two small p's are the only ones that are going to give you the white flowers. This is the 3-to-1 ratio. We can also use this as a way to determine an unknown genotype. Before we look at this example, just a couple things I want to go through in vocabulary. An individual that is homozygous is going to be an organism that has two pairs of identical alleles for a character. One that is heterozygous has two different alleles for the character. The phenotype is the organism's appearance, the type of traits you observe. The genotype is their genetic makeup. In this example here, we're going to do a test cross. We have an unknown genotype, and we're going to cross that with the recessive homozygote. This will allow us to be able to see the genotype of the organism. So here we have purple flowers. They can be purple with two big P's or with a big P and a small p. We'll cross it with the white flowers that have to be two small p's. When we look at the offspring possibilities, if you have the purple flowers with two big P's, this is going to be your Punnett square here on the left. What it will result in is all purple flowers. If the original parent is heterozygous, it has a big P and a small p, the Punnett square is going to reveal getting about a 50-50 ratio of purple flowers to white flowers. So a test cross can be done to determine the genotype of the parent. Test crosses are going to be something that can be used in plants. They are not real practical in the animal world. We call these monohybrids, where the parents were going to be heterozygous for one particular character being followed in the cross. So a monohybrid cross just looks at a cross between an organism that's heterozygous for a single character. You can do dihybrid crosses where you can look at more than one character at a time as well. The second law of inheritance was actually identified by following two characters at the same time. So dihybrids are F1 plants that are heterozygous for the two characters being followed on the cross. In this example, we're going to look at the seeds, yellow and round, big Y, big R, small r. With the dihybrid cross between the F1 hybrids, your F2 generation is going to get a ratio of 9:3 to 3:1. This indicates the independent assortment of the alleles. The law of independent assortment says that each pair of alleles segregates independently of each other during gamete formation. This is only going to apply to genes that are located on different chromosomes that are not homologous. If they're on the same chromosome, they're most likely going to separate together. The genes are going to do exactly what the chromosomes do. You have the independent assortment of the chromosomes, the genes are located on the chromosomes, so they go along for the ride. Looking at this dihybrid cross, we start with a true breeding yellow and round, and a true breeding green and wrinkled. The gametes for the yellow and round are big Y, big R. For the green and wrinkled are small y, small r. In the F1 generation, all of the offspring are going to have a big Y, big R, small r. Using independent assortment, we can create a set of all the possible gametes for the eggs and a set of all of the possible gametes for the sperm. The easy way to figure these out is if you look at using what in math is called the FOIL method. If you look at the first of each letter, big Y, big R, that's going to be one of the possible gametes. If you look at the outer two letters, big Y, small r, that's the second possible letter. The inner two letters, small y, big R, third possible gamete. The last of each letter, small y, small r, the last possible gamete. So you're going to have the same gametes for the egg and for the sperm. When you fill in the box, you're going to put all four letters in the box. So each box should get 2 Y's, 2 R's. They represent your gametes, so a person should have 2 alleles for each of the possible characters. When you look at this, you get the ratio of 9 to 3 to 3 to 1. The nice thing about Punnett squares is they are relatively simple to do, so that saves you from having to memorize these ratios. So Mendelian inheritance will follow the laws of probability of math. Probability is going to range from 0 to 1. Events that are certain to happen are going to have a probability of 1. Events that are certain not to happen are going to have a probability of 0. Everything else will be something in between, and the probability of all the possible outcomes has to add up to 1. With the multiplication rule, this is used to determine the probability of 2 independent events occurring together. We multiply the probability of one event by the probability of the other event. When we're looking at the probability of getting both of these traits together, if you have a 1 in 2 probability for one and a 1 in 2 probability for the other, the odds of getting both of them together are 1 in 4. This is the one you would probably use more often if you were looking at traits in Mendelian inheritance. The addition rule, this is your probability that any one of 2 or more mutually exclusive events will occur. This is going to be done by adding their individual probabilities. So probability for one way of obtaining the F2 heterozygote, the dominant allele from the egg and the recessive allele from the sperm is 1 in 4. The probability for the other possible way is also 1 in 4. By using the addition rule, the F2 heterozygote will have a 1/4 plus 1/4 chance, which is 1/2. If you're doing 1 or 2 traits that you're looking at, it's usually easier just to do the Punnett square. If you start looking at multiple characters, then it becomes much easier to use the probability rules. There are several examples here where you can see using the multiplication rule to get the number of recessive traits in these organisms. Sometimes the inheritance patterns are a little bit more complex than what is predicted by simple Mendelian genetics. Some traits are not going to be completely dominant or recessive. When a gene has more than 2 alleles, or when a single gene produces multiple phenotypes, it won't necessarily fit into this picture. If we look at complete dominance, that's what we've seen with the dominant or recessive relationships. Your F1 offspring always looks like one of the 2 parental varieties because one allele in the pair dominates over the other. When we have incomplete dominance, your F1 hybrids are going to have a phenotype that's somewhere between the 2 parental varieties. In this example, we're looking at these red and white flowers. Here you're going to see a superscript. So the character we're looking at is color, and the variation here is red. So this is indicated with a CR, CR for the red flower. All of their gametes will be a CR. For the white flower, it's CW, CW. All of their gametes are CW. In the F1 generation, you get all pink flowers. A pink flower is going to have a CR, CW. The possible gametes from this pink flower, when we do self-pollination on the F1 generation, is going to be half of them being CR, half are CW. When we look at this F2 generation Punnett square, here you're going to get a 1 to 2 to 1 ratio. 1 that is red, 2 that are pink, and 1 that is white. With codominance, you have 2 alleles that each affect the phenotype in separate distinguishable ways. Human blood type is a classic example here. When we look at the human blood types, we will typically use I as your base letter, and then you'll have the superscript that will be A or B for the marker on the cell. So when we look at the allele for blood type A, these cells have marker A present on them. The genotype can be IA or IA with a little I. Marker B is going to be coded with IB. The genotype will be IB or IB little I. The little I by itself has no marker on the cell. So you'll still have the marker if you have an A or a B marker present that will determine the type of blood cell. So that's how you can have IAIA or IA little I are both going to code for blood type A. If you have IA from one parent and IB from the other, you're going to have blood type AB. If you get little I from both parents, there's not going to be any markers on the cell. So we refer to this as type O. Type O means no marker. When we have dominant alleles, they are seen in the phenotype. It does not interact or suppress the recessive allele here. We look at Tay-Sachs disease. Here, brain cells cannot metabolize certain lipids because an enzyme doesn't work properly. This allows the lipids to accumulate, leads to brain seizures, other neurological degeneration, and eventually death. When we look at the organism, a child with 2 copies of the allele will have the disease. It qualifies as recessive. If you look at the activity level, the level is intermediate in the heterozygous individual because of incomplete dominance at a biochemical level. They're going to produce some normal enzyme cells, some cells without normal enzyme. At the molecular level, the heterozygous individual produces an equal number of normal cells and an equal number of dysfunctional enzyme cells. You always want to go back and look at the genetic relationship before determining is something going to be dominant or recessive or codominant. With multiple alleles, we see most genes actually do exist in more than 2 allelic forms. For example, human blood types with the A, B, and O alleles. With pleiotropy, most genes are going to have multiple phenotypic effects. In humans, the pleiotropic alleles will be responsible for multiple symptoms associated with certain hereditary diseases. This is seen with cystic fibrosis or sickle cell disease. If the gene is going to code for a protein that's seen in multiple places in the body, you will have multiple symptoms. For example, with cystic fibrosis, it's going to lead to thickening of the mucus. Mucus is produced in several areas of the body. So a person with one genetic variation would actually end up with multiple symptoms. With epistasis, you have phenotypic expression of a gene at one location, alter that at a second location. Classic example here is with lab dogs. A black coat is dominant to the brown coat, so big B is going to code for the black coat, small b for the brown coat. Those are your possible alleles. In order to get a chocolate lab, you have to have 2 small b's. There's a second gene that's going to determine whether or not they're going to deposit the pigment in their hair. The dominant allele says deposit pigment. The small e is going to say don't deposit pigment. So if a lab is BB, it's carrying the genes for a brown coat. If the second locus is EE, they're going to end up with a yellow coat because even though they're carrying the gene to make the brown pigment, the gene is saying don't deposit the pigment. The gene for the pigment deposit is going to be epistatic to the gene that codes for the black or brown pigment. If you look at the yellow labs here, they all are going to have 2 small e's. So if you breed 2 yellow labs together, there's no way of getting any other color because none of them will deposit any pigment. There's no big e's in the mix to be passed on to future generations. With polygenic inheritance, you have the additive effect of 2 or more genes on a single phenotypic character. This is going to be the converse of pleiotropy. With quantitative characters, here it's impossible to determine the exact character because they're going to vary in the population in gradations along the continuum. Human skin color and height are an example of these. If you breed a person who is tall with a person who is really short, you could get a tall person, a short person, or anything in the middle. When we look at the role of nature versus nurture, the environment definitely has an impact on the phenotype. This provides even more exceptions to Mendelian genetics when we look at the phenotype for a character that depends on the environment as well as the genotype. The norm of reaction refers to a genotype that's generally not associated with a strictly defined phenotype, but instead you see a range of phenotypic possibilities due to the influences from the environment. With multifactorial characters, you have many factors, both genetic and environmental, that are going to end up influencing the phenotype. So genotype and phenotype also refer to an organism in its entirety, not just specific alleles. In an integrative view of phenotype, it reflects the overall genotype and the unique environmental history. So test crosses work great in plants or in bacterial cells. However, they're really not necessary in bacterial cells because you don't generally mix up the genetic information, they're clones. In animal cells, test crosses are not going to be very practical. You're going to have organisms that you have to take care of in there. So instead, with humans, what we'll look at is a pedigree. The human generation span is just too long for genetic research when it's about 20 years. The pedigree is going to look at a collection of formation of the family history for a particular trait. It is definitely a lot easier to look at a pedigree just looking at one trait, and you'll look at the traits of the parents and the children across the generations. These can help you determine the probability of having a future child with a particular genotype or phenotype. In this example here, we're looking at a widow's peak. In this third generation, we have two sisters. The key to this, you've got squares that represent males, rounds represent females. Affected individuals are going to have their circles or squares colored in. If they're unaffected, they're going to be blank. This is a very simple pedigree here, so there are ways of identifying those who are characters. When you have a mating, you're going to have a line drawn between the two individuals. When you're looking at your offspring, the line is going to come down from the top. It will come down from the parents. Siblings do not have the line in between them. Their connection is going to come down directly from the parents. So here with the two sisters, we have one who has no widow's peak. She's going to have two small w's because the widow's peak is the dominant trait. To not have one, she needs to have the recessive trait on both of her alleles. The other sister, we're not sure what her genetic makeup is. So she could have two capital W's or two small w's. When we look at their parents, both parents are going to have widow's peaks. But we know that the parents also produced an offspring and had to be carrying the small W gene in order to have an offspring with no widow's peak. So they're both going to be heterozygous. Looking at mom here, mom had a sister that did not have a widow's peak. When we look at grandparents on mom's side, grandpa did not have a widow's peak, so he has two small w's. Grandma had a widow's peak, but she also had an offspring that did not have one, so she has to be heterozygous as well. When we look on dad's side of the family, dad has a widow's peak, but is going to be heterozygous because he has an offspring without a widow's peak. He has two brothers that don't have widow's peaks and a sister that does have a widow's peak. When we look at grandma and grandpa on that side, grandma has no widow's peak, so she's going to be homozygous recessive. Grandpa has a widow's peak, but because there are offspring that don't have a widow's peak, grandpa had to carry the small w. When you look at the possible alleles available to the sister with the widow's peak, she was going to have to be heterozygous and inherit a small w from her mother. So when you do these pedigrees, a lot of times you'll write out the relationship, color in who's affected, who's not affected, and then you have to take the steps one at a time, looking back and forth between the generations to determine the genetic makeup. Recessive disorders, the heterozygous individual is typically normal in phenotype. They're going to have one normal allele, and it will usually produce enough of the specific protein so the disorder only shows up in the homozygous individual with the recessive alleles. The carriers are going to be the heterozygotes. They're phenotypically normal, but they can transmit the recessive alleles to their offspring. What we see with genetic disorders is they're not evenly distributed among all groups of people. This is due to the fact that we have different genetic histories and times when we were more geographically and genetically isolated. Even though we all have access to hopping on an airplane and finding a mate on the other side of the planet, we're still most likely to find one that is close to home. However, you don't want to find one that is too close to home. Consanguineous matings, these are inbreeding. It indicates a pedigree by double lines. Here, your probability of passing on recessive traits increases greatly. This is a problem with domesticated animals. They are not supposed to be inbred, but it does happen. Cystic fibrosis, this is the most common lethal genetic disease in the U.S. It affects 1 in 2,500 individuals of European descent. It's more rare in other groups. 4% of European descent people are carriers for this. The normal allele codes for this Fro protein that's going to be involved with transporting chloride ions. When this is defective in the recessive homozygous, they're going to have mucus that is thicker and stickier. This will be abnormal and build up in places like the lungs, the gastrointestinal tract, and any other organs that have mucus. This leads to poor absorption of nutrients, a lot of lung infections, chronic bronchitis. Today, about half of those with cystic fibrosis will survive into their 20s and 30s. So the lifespan is improving. We have a lot better supportive treatments for them now. With sickle cell disease, this is the most common inherited disorder in people with African descent. It's in 1 in 400 African Americans. You substitute just a single amino acid in the hemoglobin. The homozygous individual will have all red blood cells abnormal. With low oxygen content, the red blood cells will aggregate into sickle shapes and clump and clog the small vessels. This can lead to other symptoms, including weakness, pain, damage, and paralysis. The heterozygous carriers have what we call sickle cell trait. They're usually healthy, but they can manifest the symptoms with prolonged reduced oxygen. 1 in 10 African Americans have sickle cell trait. The heterozygotes individuals have a reduced frequency and intensity of malarial attacks. So this has been a real advantage in subtropical Africa where malaria is common, and has helped to increase the frequency of this gene being carried in that population. So dominantly inherited disorders, achondroplasia is a form of dwarfism that affects 1 in 25,000 people. Here, the heterozygous individuals have the dwarf phenotype. Typically, dominant alleles for lethal diseases are much less common than recessive alleles, and that's because the lethal effects tend to usually kill the afflicted person before they're able to reproduce and not get passed on as frequently. An exception to this is Huntington's disease. It's a late onset lethal disease that's going to involve degeneration of the nervous system. There's no phenotypic effects until the person is 35 to 45 years old. And at that point, most people have usually already had some of their offspring. This affects 1 in 10,000 in the US. They can actually do genetic testing for this now. With multifactorial disorders, you have a lot of people that are susceptible to diseases on a multifactorial basis. There's a genetic component as well as a significant environmental influence. Some examples include heart disease, diabetes, cancer, alcoholism, schizophrenia, bipolar disorder. There is likely many others that are going to be in this category as well. Genetic testing and counseling is available. There's a lot of genetic tests that are available to identify heterozygous carriers. You may be able to assess the risk prior to conceiving a child. However, it always does raise ethical questions, particularly who gets to know the information. Also, concerns with privacy. We do have some ways of doing fetal testing. With amniocentesis, this is usually done during weeks 14 to 16. About 10 milliliters of amniotic fluid is extracted, And some disorders are able to be detected by the presence of certain molecules in the fluid. Others can be tested in DNA from the fetal cells that are sloughed off in the fluid. This allows you to be able to do a karyotype. With chorionic villi sampling, a tube is inserted through the cervix and suctions out a small sample of the placenta. This is done a little bit earlier, usually in weeks 8 to 10. Here you get cells that are derived from the fetus and can do a karyotyping and genetic testing. Ultrasound is non-invasive. This allows for screening of anatomical abnormalities. This can be done to determine the sex of the child, and oftentimes people will do that to determine the sex of the child, so it's done routinely on many pregnancies. With fetoscopy, fiber optic viewing scope is inserted into the uterus. This will allow you to actually view the fetus. With blood tests, you can isolate some fetal cells and DNA that have escaped into the mother's blood. You get very few cells, but they can be cultured and tested. You can also do some screening after birth with newborns. Some things can be detected with simple chemical tests, such as PKU, phenylketonuria. This affects 1 in 10,000 to 1 in 15,000. These individuals cannot process phenylalanine. They produce toxic byproducts when they consume it, and they can accumulate, causing mental damage. This can be treated by having the individual be aware and choose to consume a special diet.